All right, guys, so on this test for Monday, first of all, some things that you ought to already know. If you don't, you haven't been paying attention. But if you want to do well on the test, you need to look back to older videos to get this information. Make sure you know the difference between prokaryotic and eukaryotic cells. Know some examples.
Do you do the videos for quizzes too?
No. Well, the lecture videos are posted, so yeah, sort of. But not a specific one for a quiz. Make sure you know the differences between the plant and animal cells. Know what all the little organelles do. Mitochondria, chloroplast, ER, Golgi. Know what they all do. If you need help on that stuff, go back to the last couple of videos and look at them. I'm not going to take the time right now to go through that. But I am going to look at the diagram, which you guys have a copy of sitting in front of you. And here is that diagram. Let's talk just a minute about the parts on that diagram. I'm looking for something here. All right, so notice that number 1 is in both things. This is a plant cell on the left, an animal cell on the right. Number 1, right here in the plant cell. Notice it's much smaller in an animal cell. That's one of the differences between the two. What is it? It's a vacuole. Plants have that big old huge vacuole in the middle, mostly to store what? Water. Mostly water in plants. Animals might use it to store water. They might use it to store food. They might use it to store waste. They might use it to store enzymes. But that's a vacuole. Number 1's a vacuole. All right, structure number 2. Notice it's also in both places. It looks like sort of an oval, and then inside there's another little membrane. It's all curvy. What's that? What is that supposed to be? It's a mitochondria. And remember, the mitochondria is where respiration, aerobic respiration happens. It's where your cell makes what? ATP. Plants and animals both have mitochondria. All right, number 3. Where's 3? 3 are just these little bitty dots. Some of them are just sort of floating around out in the open. Some of them are on top of, this is the ER. Just one second. Both cells have them. I don't see them labeled in this plant diagram. Yeah, there they are. These little dots. What are them? Just a little bitty dots.
Ribosomes.
They're ribosomes. And remember, those are the structures in a cell that make what? Protein. Protein synthesis. Plant cells have them. Animal cells have them. All right, number 4 in your diagram is the ER. And here's some ER. Here's some ER.
Is it smooth or rough?
All right, well, they didn't differentiate on the diagram, so we're going to differentiate. If it's got all the little dots on it, it's the rough, right? If it doesn't have the little dots on it, it's the smooth. Remember, they've got slightly different things. They both help to transport things. But the smooth one also helps your body to make lipids, fats, like for the cell membrane, and it also helps to get rid of toxins. Whereas the rough one is mostly about packaging proteins and shipping proteins, moving them through the cell. Going on down the list, number 5 on your diagram. Notice it's right here on the animal cell, and it's right in here on the plant cell. That would be what? That's the membrane, the cell membrane or the plasma membrane. It's the gateway in and out of cells that can control what enters and exits the cell. Joyce, what were you going to ask?
What is the smooth again?
The smooth, what does it do?
Yeah, it doesn't help make lipids.
It helps to make lipids and get rid of toxins, waste, poisons. Yeah, you want? All right, moving on down our list. Let's look at number 6. 6 just looks like a small vacuole, which is really sort of what it is. Notice that 6 is labeled in the animal. Plant cells don't usually have these. These are lysosomes. And remember, lysosomes are used to break things down inside a cell. We said they're sort of like when we talk about factories, we said they're sort of like what part of the factory? Recycling center. They help to do what's called intracellular digestion, break things down inside a cell. They have different enzymes to break things down, but it's not enclosed like our lysosomes are. All right, number 7 on this diagram is just this white space inside the cell. That's just white. What's the white space?
Remember?
Inside the cell.
Oh, that's
Cytoplasm. It just fills up the cell, number 7. Number 8 in your diagram. 8's this whole structure here. It's this whole structure here. So that's the nucleus. Main thing, that's where the DNA is at, right? It's the control center.
What do you say from cytoplasm?
It just fills up the cell, supports the cell. Control center contains DNA. All right, number 9 is only labeled in the plant. It's this outermost part of the plant cell, and it would be what? That's the wall. Animal cells obviously don't have a cell wall. It's there for support, protection. Number 10. Number 10 should always be near the cell membrane. I don't really like the way they drew it on here. Here's number 10 on this one. Number 10 on this one is right here. Usually it's going to be closer to the membrane, and it's usually going to look like, in most diagrams, it looks like little stacks of pancakes. What would that be? If it's close to the cell membrane, it looks like little stacks of pancakes. It's a Golgi apparatus.
It looks the same thing.
It looks a lot like the ER, but the differentiation there is that Golgi is usually closer to the cell membrane. I know. That's what I'm saying. This is not a great diagram. But the rough ER is almost always really close away,
Chromosomes.
which are both close to the nucleus. Because what do they have to get to do what they got to do? What do they have to get from the nucleus? Yeah, but what do the ribosomes need from the nucleus? They need the RNA, the directions for building the proteins. So usually the ER is going to be close to the nucleus, and the Golgi is going to be close to the cell membrane, typically.
So what does the Golgi do?
Yeah. We just talked about that on the quiz a second ago. It packages proteins and helps to ship them out. Exocytosis, that was the question on the quiz. All right, number 11 is just this membrane around the nucleus. Notice it's there in the plant. It's here in the animal. It's the nuclear membrane. We've also been calling it what? Nuclear envelope. Essentially, all you need to say there is that it encloses the DNA. Yep. Nuclear envelope encloses the DNA.
You can go slower.
The main thing I want you to do right now is just label it. You should know the functions. All right, and number 12 look like little vacuoles with little stacks of pancake-like things inside. Notice they're only in the plant cell. There's a bunch of them. What would that be? That's a chloroplast. And that's where photosynthesis happens. And then the last two, you've got a nucleolus, which is this dark spot inside the nucleus. Most cells have it. It's number 13. And that's where ribosomes are made. Ribosomes are made in that little dark spot, the nucleolus. And then the very last one, it's usually around the nucleus. This is one we haven't talked a ton about. That thing. Notice it's found in the animal, not in the plant. We have a question about it on the test. Looks kind of like an X. What thing on the test, the quiz we just took? Centrioles. Animal cells have them, plants don't, and they help things like mitosis to happen, cell division. All right, so there's 3 or 4 questions that will be diagram questions on your test. Make sure you know the parts, know what they look like. All right, and then the last kind of stuff was the cell transport stuff. You saw in the quiz, there were questions directly about the definitions of these things. There will be on the test too. Diffusion is just the movement of something from left to
Well,
left.
we have to label this thing on the quiz.
There's some questions about labeling, yes. But back to what I'm saying, diffusion. Movement of a substance from high to low concentration, right? Distribute. You should already have this. Osmosis is the movement of what? Water. The movement of water. We defined it 3 different ways. We said it's the movement of water from high water concentration to low water concentration. That's the most basic way to define it. But we also defined it in terms of what was in the water, which we call what? Solutes. And if we're talking about what's in the water, it's moving from low solute concentration to high solute concentration.
Is the blue one for diffusion and the brown one for osmosis?
No, guys, you gotta listen. We're just reviewing. You don't need to be writing this down. You should already have it written. Just listen. This is just a review. Osmosis is the movement of water from high water concentration to low water concentration. Another way of defining that is to say it's the movement of water from low solute concentration to high solute concentration. And yet a third way to define it, which was a question on your quiz, is when water moves from what? How do I need to finish that up? Hypotonic to hypertonic. All 3 of those definitions say the same thing. They say it in slightly different ways. They all mean the same thing. Facilitated diffusion. The next one is diffusion with a little help. Facilitate needs the help. What kinds of things need a little help to get in and out of the cell?
Big proteins.
Big things and charged things. This is what helps them. So this is the movement of a substance from high to low concentration through a protein channel. Still moving from high to low. So it's still what kind of process? It's diffusion, but it doesn't require the cell to use what? ATP. So what's the word for that? If it doesn't require passive. What provides the energy to make it happen? Heat in the environment. Doesn't have to use its own energy. It's facilitated diffusion. Active transport's the next one on the list. This is the one that's moving things opposite of the way nature tends to want to move things. Nature wants to move things to get them even, right? So it's always moving from high to low. This is when things get moved from low concentration to high concentration. This is when a cell is trying to build up something. Trying to build up a lot of something in one place and not very much of it in another. Active means what?
Uses ATP.
Uses ATP. The cell is having to use energy to do it. It's really important in the nerves. And then these last few things, we could group all of those up under a heading: bulk flow. Bulk flow. Because all those bottom things are ways to move big amounts of stuff. The top things, we're moving atoms and molecules. The bottom, we're moving big hunks of stuff. Endocytosis, if you just break the word up, just means what? What's endo mean?
Absorbed into the cell.
Inside, cyto means cell. Inside the cell. So it's bringing big amounts of stuff into the cell. There's 2 kinds of endocytosis. Phagocytosis is when solid materials are engulfed and taken in. And pinocytosis is when dissolved things, solutes, are taken in. But they're both parts, both kinds of endocytosis. And then the last one up there, exocytosis, is moving things out. And usually what organelle is involved? The Golgi apparatus. So it's moving large amounts of substances out of the cell. One more thing. There might be some questions about the cell membrane as well in this test. What's the main component of the cell membrane?
Phospholipids.
Phospholipid, add another word there. A bilayer. A layer of phospholipids that might look like this. Where you got this little head that likes water and the tails that don't. So your phospholipid bilayer might look like this. There's 2 layers of them. What kinds of things did we say could go through that pretty easily?
Smaller and non-polar.
Yeah, small and not charged. Small and not polar. But there have to be some other things, other parts of the membrane. You might have something that looks like this. What might that be? What kinds of things? Yeah, they're stuck in there. Those are transport proteins. Hold that thought for just a second. Hello? Eric, you're being paged to the attendance office. Check it out. All right, so what I was saying, go ahead, please. Cell membrane is mostly made out of lipids, phospholipids, but there's some proteins in there. The proteins may be like channels. We'll call this one a channel protein. It helps to move big things and charge things in and out. There also might be little proteins that might look something like that. We're going to call those receptors. What does the cell membrane need to receive?
Signals.
Signals. What kind of signals?
Yeah.
What kind of chemical signals float around in your bloodstream? Hormones. Okay? Might need to pick up hormones. You might also have little parts, little proteins and sugars sticking out the surface of your cell membrane. And we call these things marker proteins. And those proteins, one of their jobs is to tell what part of your body, hey, this is me. You know, what part of your body needs to know this is me. Your immune system, so that you don't attack yourself. The last thing that I want to review here for the test is a problem like the one we just had on the quiz. And let's do one more of those. Let's say that we had a beaker, and in that beaker, let's say we had 2 molar sucrose, and we had our little baggie, and inside the bag, we have 0.5 molar sucrose. Now, I guess it was Taylor who asked a minute ago, when they give you these numbers, that's how much sugar is in there, but what's implied to be there too? Water. Okay, it's dissolved in water. That's what molarity is telling you. Is you got this much sugar dissolved in water. So here's the deal. If the sugar could move, which way would the sugar go? If the sugar could move, which way would the sugar go? If it could move, which way would it go? Look.
Where's the sugar?
2 molar sucrose outside, 0.5 molar inside.
It would go all the way.
It would go in, right? Because the sugar wants to move in from high to low. But what do we say about the sugar?
You're too big.
It's too big to get through the holes, so it can't. So the sugar can't move, so what's going to move? The water. And which way is the water going to go in this example?
Inside.
It's going to go out. Because how does water move in terms of the solute? From where there's less solute to where there's more solute. So the water is going to go from in the bag to the outside. And this bag, this cell, if you think of it as a cell, it's going to shrivel up. Those are the kinds of things you need to know for the test. Take some time, study. This little video clip will be posted. Make sure you study for your test.